So, welcome to the first lecture of the first week of the course Functional Genomics. So, in today's lecture we are going to look into the growth of the field called genomics. How did really this particular field came into existence, and who are the main people who contributed to the growth of this course, or rather the field. And so this is going to be a bit history, but it's very important and interesting. So I thought I would share with you. If you look into our human body, then you know that the smallest unit in terms of functionality, you can call it as a cell. And cell, as all of you know, has got chromosomes, which has got DNA, and that's what the genetic material, which has all the signal as to how the cell has to function, specific to that tissue that it is present in, and how that tissue contributes to the function of the system that it has, be it skin, be it digestive tract, and then how all this system gets integrated in terms of function to be a successful organism. So, the basic information that a cell needs, or the tissue requires, or the organism requires, is present in your DNA. That is our belief, but we do not know really how and what way the sequence information is understood and the cell is able to perform its function. So, one of the major questions that most of the scientists have been asking since centuries back is to understand how the genetic material, or what is the fundamental information that is present in the cell, and how that information is retrieved and executed. So, that you can go back in history, and we will look into some landmark discoveries as to how we understood. So, if you look into the chromosome as is shown in the slide, you can pull out the DNA from the chromosome. You know that from the top to the bottom, what you call as the telomeres, the two ends of the chromosome, there is one double helix that run all the way. So, that is what you call as a DNA. And of course, a small segment of the DNA constitutes a functional part of a gene. So, this gene has all the sequences that constitute the gene in terms of its function. It includes, for example, the region that gives you the signal as to where the transcription should begin, where it should stop, and which sequence the RNA should be translated, meaning copied for making amino acids to form peptide and so on. But if you look in the history, that the first time somebody really talked about that there are certain factors that are there in your cell that gives you a particular phenotype. When I say phenotype, it could be any of the changes that you see in a body. For example, whether you are able to roll tongue, or you are unable to roll tongue, or the way your hair texture is, whether it is straight or curly, or for example, you have free earlobe or it is fused. Sometimes it could be your height, and there are many other such phenotypes that are regulated by your DNA. So, what is that in your genetic material that really regulate? This understanding comes from one of the pioneering studies done by this monk called Gregor Mendel. He looked at plants, as most of you would have studied, onto some of such contrasting characters like height of the plant, the color of the flower, the size or the shape of the seed, and so on, and try to understand whether whatever factor that determine as to what should be the phenotype, be it the height of the plant or color of the flower or the shape of the seed, whether these are transmitted from one generation to the other and how they are transmitted. He looked at, he did some really amazing experiments. You have to go back and study the original literature. You can understand how beautifully he selected the plant, how beautifully he selected the phenotype, and came up with hypothesis then he called us, there are 3 hypotheses which talks about segregation, dominance, and what is called as independent assortment, which we call them as laws now, because every gene obeys whatever hypothesis that he proposed, therefore we call them as laws. So that was the birth of genetics, generally people consider, because that is the one that really gave the fundamental understanding as to how the genes can regulate without, Mendel of course did not know that there is a DNA, there is a gene, but he said these are some factors, and beautifully has shown. And still if you look into the human population or animal or plant, and this is what you see, all the genes and their phenotype obey the 3 hypotheses that Mendel proposed, and that is what we call them as laws. So that is the pioneering discovery, and that led to a kind of a concept that there is something there in your cell that is transmitted to the next generation that governs your character. Almost at the same time, somewhat overlapping, is another person who came called Fleming. Fleming again, he looked at plants, but not like Rigger Mendel. Mendel looked at plants as a phenotype. He looked at how the plants look like, what is the height, what is the color, what is the seed, and he really did wonderful experiments. He crossed them, looked at the second generation, third generation, came up with the laws. But this gentleman, Fleming, he did phenomenal work. He looked at the cells of the plants. Okay? These cells as to how do they divide. So what you see on the right side in the PowerPoint slide is one of the drawings that he made after looking at the cells that are dividing. So you can, for example, those who have done biology in your school, you must have used the onion root tip to look at how the cells divide, mainly the mitosis. So basically he also did very similar kind of, he is the one probably started with such kind of analysis, and they drew all these different stages, and you can see beautifully the chromosomes that are forming in the metaphase and how the cell is about to divide, and you have this cells about to divide and so on. And he came up with the theory as to, you call them as the concept that we are talking about now, chromosome and chromatin, meaning the chromosome is a distinct entity which is different from the other. Chromatin is what you call as the DNA with the protein that forms the compact structure. Now this is what he drew, and he called them as chromosome and chromatin. The very concept that you have something like this which becomes compact, and when the cell divides they are sorted, has come from this gentleman Fleming. So that is his phenomenal contribution. You can see when possibly it could have done. And then came another person, Boveri. You can again phenomenal. He basically looked into the chromosomes, and he looked into what is called as meiosis, how cells divide in the germ cells that makes gametes. So he looked into the reductional division, whereas Fleming looked at your mitosis, the normal cell division. So Boveri really gave a kind of explanation as to what Mendel has proposed. That is, that you have different forms of the genes, what you call as alleles. For example, a gene could determine your height. So you may have 2 variants of the gene, one that gives you tall phenotype, the other one gives you somewhat short phenotype, and these 2 get separated during what is called as gametogenesis. Therefore, if you cross a plant that is tall, but having one allele that is giving the phenotype that is not that tall, and cross it with another plant which is not tall, then you would expect in the next generation 50% of them not being so tall, right. So that means that chromosomes, the 2 alleles are sorted into 2 gametes when the cell undergo meiosis, and basically looked into the meiotic process, and he has shown that the chromosomes get sorted and they become kind of a reductional division happens, and that is something that his contribution, again it is a very major contribution in terms of understanding how the genetic material, then they do not know that it was a genetic material, but whatever it is, it gets sorted in the cell. But almost at the same time, these all three people, Mendel, Fleming and Boveri, they all from European origin, and they were working from the Europe. But at the same time, Walter Sutton, he is an American who also was looking at the same phenomenon as to how the chromosomes are sorted in meiotic cell. And what is shown on the right side is his one observation. He basically has shown that a cell which is undergoing meiosis at the end of the cell division, the number of chromosomes become half. So, you can see that what he call as a somatic series, the chromosomes he named by then, and then you say somatic series, if it is 2, in the reduced series it is half of it. So, he has basically has shown that how the reductional division takes place, and then he spoke about combination in gametes and combination in zygote when this two gametes, the sperm and ovum, when they fuse together to form what you call a zygote, how they again restore the original number that was seen there. So, basically he looked at all these things, and that is how he has shown that the meiotic cells undergo what is called as reductional division. That is an amazing discovery. So, these are all independent. Boveri and Sutton have done independent observation, but eventually turn out to be the same. What is interesting here is that what kind of organisms they have used to understand these phenomena, whether it is mitosis or meiosis, which pretty much universal. So, whether it is a plant, animal, human, you find the cell divide to grow or to repair the damaged part by mitosis, right. So, there the DNA replicates, meaning makes another copy, and then eventually cell divides, you restore what is the normal number of chromosomes. But it was very difficult, not until 1960s people have not really looked into the human system because it is very difficult. So, they have used a variety of different organisms to understand, like for example, Fleming, I said he looked into plants, and because easily one can do the preparation and see the chromosome, that is why he named it as chromosome and chromatin. But Boveri and Sutton, they looked at meiosis, which is very difficult. So, they went and looked at animal systems. So, much of our understanding in the early years for mitosis have come from plants, whereas for meiosis have come from animals, although such cell division do happen in the plants as well when they make the sperm. Boveri used a worm called Ascaris, he looked into that and looked at the meiosis process, whereas Sutton looked into grasshopper, obviously an insect, American insect. And in these species, the tissue that forms the gamete, the testis, is soft, one can dissect and looked at, make it thin layer under a microscope, and then you can stain for the chromosome and look at, and that is how they really came up with these discoveries, right. So, what is generally spoken as a descriptive science in biology have come from such kind of observation. They did nothing but looked at different snapshots of event that took place when you fix the cell and observed them and tried to correlate as to what could possibly the sequence, and they came up with the discovery which is amazing. So, in science there is nothing called descriptive or analytical experimental. Science is science, that you guys should remember when you study anything. So, that is the system, like Ascaris and grasshopper really contributed to the understanding of the concept that even talk about in the humans today, whether it is disorders or reproduction or normal growth, whatever observation they have made in any of the system really helped us in understanding what goes on in our body as well. So, the fifth person I am going to introduce is Bettison. So, he is again the one who made or termed the field called genetics. He is the one who gave this term, and he is the one who really understood what Gregor Mendel has done. By then, until then, Mendel's contributions were not known. He has done that and he recorded and left, but nobody really looked into the implications. Some of them who looked into felt like what he has done is absurd, is not explaining anything, it may not be applicable to humans and animals and so on. But it was Bettison who really looked into Mendel's data, and then he developed the field. He coined the field term called genetics, and then he said what Mendel did applicable to each and every organism. So, he pretty much popularized what Mendel has done. Then people took note of Mendel's contribution when Mendel was no longer living. So, that is the legendary work that he has done. So, then there are people who really try to look into variety of model system to understand, because depending on what question you are asking, you need to have appropriate model system to understand. That is when Morgan came, an American geneticist, and he started using the most famous model system that we have now, Drosophila. So, he basically went with what Sutton did, using insects as a model, but he found Drosophila to be much better for a simpler reason. One, it has a shorter lifespan, so you can cross them to have the next generation and so on, because genetics cannot be done without looking into the generations. And two, the organism has got a fewer number of chromosomes. So, he basically wanted organism that has fewer number of chromosomes, therefore, whatever characters that are linked to the chromosomes are fewer, therefore, you will be able to understand better. So, what is shown on the top here in the slide on your right side is chromosome schematic of the chromosome from a female and a male Drosophila, which of course, you have this like human, you have autosomes that are depicted on either side, and then you have the sex chromosome, which is XX or XY. So, he has used this Drosophila to understand the chromosomes, and he is the one who really established what you call as genetic linkage, meaning there are different segments of a chromosome, and these segments represent a character, like what Mendel's said. It could be a gene that determines your height, or it could be a gene that determines your any other phenotype. For example, in plants it could be the color of the plant, it could be the shape of the seed, and so on. And he said that they are physically connected to each other. And then he also said, though they are physically connected, when you have a pair of chromosomes which are homologous, meaning identical but about to be separated during the formation of the gametes, each of the homolog could have what is called as a variant of the gene, the other form of a given gene, what you call as allele, and there could be shuffling take place, meaning there could be exchange of DNA material from one homolog to the other, and that we call as crossing over. So, he hypothesized everything by only looking at the chromosomes in the fly, and then during different generations, and then looking at how the fly looked like. So, like Mendel, he also selected flies that have variants in terms of how do they look like. For example, it could be the color of the eye, it could be the shape of the wing, it could be the size of the fly itself, their color, body color, and there are many different patterns that we will discuss a little later. So, he has used that to show that all these phenotypes, just like Mendel did, can run through in families, meaning different generations, and they are linked to the chromosome, some of them in the same chromosome. Therefore, you can show that more often they are transmitted together, right. So, this is the major difference between Mendel and Morgan, because Mendel, whatever character he took, it so happened, or he has selected those characters that are present on different chromosomes. That is why his law, the third law, what he calls independent assortment, applies, because that law applies to genes who govern characters. The genes are present in different chromosomes, that is why they are sorted independent of each other. So, this law does not apply to genes present on the same chromosome. They are, because they would, if they are closer to each other, they are going to go together often. They are separated, there may be crossover, but it cannot obey the law that he has given 9:3:3:1. That applies only if they are present on different chromosomes. So, that is where Morgan has really contributed to show the linkage, and he gave what is called as a genetic distance between genes by looking into the recombination frequency and so on. And this model, what he developed, became very famous model, and most of the genetics concepts and developmental biology, what you call, how the genes regulate development, how come from this beautiful system. That is the contribution of Morgan. So, they not only contributed in terms of developing model, but they also trained their students to ask more deeper fundamental questions, some of them which they could not test. One of his students, Morgan's students, later went on to look at some other aspect, but the concept, the very concept what is called as chromosome theory of inheritance, meaning your cells do have a genetic material which you call as chromosome, and these chromosomes are transmitted to the next generation, and chromosomes have certain segments which dictate what would be your phenotype. We call as chromosome theory of inheritance, and this is called as theory then because it is purely observational. There is no evidence to show indeed that is the case, because they did not know what the chromosomes are made up of and what really dictates as to which phenotype should be there. They were not clear about that, but they made this theory. But now certainly the chromosomes are the basic material, and these people that you see in the screen are the major contributors. There are many others who I did not discuss, but these people are the major contributors for our understanding as to how the chromosomes contribute in terms of your phenotype that goes from one generation to the other. So, that is a major contribution. As I told you, these contributions are not only in the science, but they also trained people. So, that is where Morgan's legacy, even today you see him, because he has trained students who took up newer challenges and developed new models depending on what kind of questions they are asking, and that led to discovery of genes and DNA and so on. So, let us look in here. For example, what is shown here in the cartoon is, so far we spoke about the chromosome and the chromosome theory of inheritance, but if you pull out the DNA, the DNA, as I said, as we know now, has got segments which are the functional unit what is called as gene. So, a chromosome could have thousands and thousands of genes. Now, the question is, how did we really know that the chromosome is made up of DNA? Because whatever the previous section that we have seen, whether it is Boveri or Sutton or Morgan, they looked at something that are there inside the cell that gives you particular staining property, which becomes compact, segregating to the daughter two cells that are dividing, but they name it as chromosome without knowing what it is. It is like you are seeing something, but without knowing what it is. Similar way, someone really, there are many scientists who looked into what is this chromosome, what is the chemical composition of the chromosome, and how that chemical moiety or biomolecule that is present there really gives the information as to what phenotype you should have. So, that has come from, again, several scientists who came after Morgan. Two of them are these people, George Wells and Edward Lowry. These are the people who gave the theory called one gene, one enzyme hypothesis. If you look into the textbook, even biochemistry textbook or genetics, this is how it will be called, and it is very, very famous hypothesis, because they said that there are certain segments in the chromosome that gives an information as to what protein should be made, and so happened here, obviously that protein is an enzyme that they looked at. Both of them are students of Morgan. Both of them were trained in Drosophila, and they know all the genetics that we discussed some time back, but they felt you have to go deeper than looking into just chromosomes and looking at next generation, what is the phenotype. We have to understand how whatever that is there in the chromosome that dictates as to how the organism, the fly for example, behaves or what phenotype it has. So, they found that system, the model system, is good for genetics, meaning looking at the chromosome and segregation pattern, but it is not so good in terms of understanding what kind of information that the chromosomes give. So, with the available tools that they had, they could not go deeper. So, they looked at some system where they can go and study, for example, proteins, meaning enzymes, their functions and so on. At the same time, they use the same technique that Morgan used, that is creating mutations. What he did was he looked into different flies wherein there was natural variation. That variation resulted in different phenotype, whether it is eye color or the body structure or the wing shape and so on. So, they went for a system which of course can be, which you can create certain changes whatever the genetic material, and then look at the phenotype, and then now go at the protein and study the protein. So, they have selected a system called Neurospora. It's a fungus mold, and there are forms what you call as haploid, meaning just like a gamete, like just one copy of the genome, or diploid, which is 2 copies of the genome and so on. And you can create mutations and then look into isolate, for example, you can grow them and you can extract protein from there and understand the function of the protein. So, they went and looked at the biochemistry, enzyme activity and so on, combining genetics with biochemistry, and they came up with clear understanding that there are certain phenotypes that are caused due to enzyme deficiency, and these are linked, just it could be one gene, which that's by then they have given this term called gene, a segment which possibly gives the information as to what function it should do. Here it is a protein that has an enzyme activity. So, they developed this particular model system to understand the protein. So, protein became much easier for people to handle, still DNA is far away. So, they started studying protein. In fact, we will be talking about a little later, we were able to sequence the protein much earlier than we could sequence the DNA. So, much of our understanding on the protein have come much before the DNA sequence, DNA structure was, or even the RNA structure or sequence was solved. In that way, they were pioneering in terms of connecting the protein with the gene. Then we have set of people. Again, you must have studied this in the textbook. This is Avery, MacLeod, and McCarthy. These 3 gentlemen together, proven that the DNA is the genetic material. Although the protein, Wells and Lowry did say that protein connects to the DNA or gene without knowing what is the gene is. But these are the 3 gentlemen who really established that DNA is a genetic material. By then we know that all the living system that has cells has got 3 major biomolecules, the protein, nucleic acid and the lipid, and then they are able to carbohydrate and so on, and they were able to show that it is not the protein, but it is the DNA that gives the phenotype. So, you must have studied about how they have used the microbes that are virulent, which can kill the animal, and they are able to separate the protein or labels protein and DNA, mixture them, and then able to show that the DNA is the one that gives the phenotype, the virulence, right. So, you can look into that. I am not going into the detail, but most of the textbook really talk about it. So, what you will see now, there is a shift. So, when the theory was about the chromosome, we looked at there are mostly plants and insects being used to understand how the chromosome functions. But now you will find all of a sudden people are going to the new system, because the question that you ask is very, very different. You find that Neurospora, the fungus, was used, and then you will find again quickly that these are microbes that are coming, in fact, from the concept called as molecular biology, where you really look into the molecule, whether it is whether it's DNA, RNA, or protein. All these understandings have come from microbes, because they are much easier to handle and grow them in the culture, and then the complexity is less. So, you will find now that really the understanding of the microbes led to the field what is called as molecular biology. That led to the genomics. I will come back a little later. Of course, this is considered to be the landmark discovery. There are controversies as well, but it is Watson and Crick in 1953 who proposed a model for the DNA. So, although the Avery, MacLeod, and McCarthy, they did say that DNA is a genetic material, the structure and how that particular molecule can function as a genetic material was not understood until Watson and Crick they proposed. It's a model. That is what shown here. This is on the right side of the screen. What you see is that seminal paper published in Nature, just one page, proposed model for the DNA double helix. It was a model. Now you know that it's no longer a model, that is indeed the structure, and that basically gave an understanding as to how the DNA as a genetic material can function, because it is double helix, it is anti-parallel, reverse complementarity. All these things help the DNA to function as genetic material, because one of the strands can serve as a template to make a new strand, and likewise the new strand can be a DNA or RNA. If it is RNA, it's a perfect copy of what the sequence is there, and that can go and give the signal as to what protein it has to make. So in that way, it was considered to be a landmark discovery, and all the textbook describe as to how important that discovery is. So that is the birth of what you call as molecular biology. So the moment we know that there are 4 bases and they complementary to each other and they base pair and form a structure like this, anti-parallel, and then people started studying into how the DNA is copied into another copy of the DNA or to RNA, and how really that functions as a biomolecule. So what we are going to discuss now is about 40 years of history, where what you call as a molecular biology era. So that basically laid the foundation for what you call as genomics now. So without this revolution of what is called as molecular biology, we wouldn't have reached what you see as a genomics. Let's see how this journey began. It's all started with Kornberg, who looked at, again, this what we are talking about from 1956 to 1990 is going to be, most of them are studies based on microbes, whether it's a phage, whether it's a bacteria, and so on, E. coli and so on, or yeast and so on. So Kornberg is the one who really looked into the process of DNA being copied. So he looked at an enzyme which reads one of the strands and then makes a copy of itself. It is a seminal discovery again, because in terms of how the DNA can be copied, because that is important for the cell to divide. The cell has to divide, it has to make a copy of the entire genome and then put them into 2 daughter cells. That's how you grow. So that is fundamental for any living system, whether you are multicellular or unicellular. So it is a seminal discovery that started with that. Then we came Jacob and Monod, because these are the people who really shown how DNA is copied into what is called as messenger RNA or mRNA, and what you study in textbook, what is called as Jacob-Monod pathway, the lac operon and so on, are the discoveries which talks about even how the gene is organized and what are the different elements that control the function of the gene, and how mRNA is made, copied, and how the RNA is important for whatever phenotype you see. See, that's very seminal discovery that came in 1961. Then we have this gentleman, Marshall Nirenberg, again an American scientist, who deciphered the genetic code, the triplet, the 3 bases together gives a code as to what amino acid should be made when the RNA being translated, meaning decoded. And later, of course, you have our scientist of Indian origin, Khurana, really helped in solving all the that different codons that you have in the table. They are the people who solved it, and but the seminal contribution that 3 bases together form a codon has come from the study of Nirenberg. In 1970, there was another landmark. Here they looked at another enzyme which copies the RNA into DNA, what you call as reverse transcriptase. This reverse transcriptase came from the group Baltimore, Dalbecco and Timin. They contributed in understanding a special polymerase which copies the RNA into DNA. That really helped us to understand how the virus functions and how they are able to infect other cells, the virus that have only RNA as a genome, and that is the discovery part. But that really helped us to make several tools. For example, we understand now what are the RNA that we have, we convert them into DNA. That is doable only by this particular enzyme called reverse transcriptase. In that way, that is seminal. Then came in 70, Hamilton Smith discovered another enzyme again from microbe. These are called as restriction enzymes. You know now that these are the enzymes that can identify a unique set of sequence in the DNA and make a cut there. And regardless how much enzyme you add, these enzymes would cut only if such sequence is present, not otherwise. So that really revolutionized the way we understand DNA, because the so-called recombinant DNA, meaning combining two DNA from two different source, is possible because of this enzyme, because now you can cut the DNA with the enzyme and join them, stitch them together. So that again is a fantastic achievement, and that led to, for the first time, Paul Berg made a DNA from two different sources, what you call as rDNA or recombinant DNA. That is again a seminal discovery, that you can stitch the DNA together and form a new synthetic DNA having different source, is something amazing, because that is what we use every day now in recombinant DNA technology or molecular biology, genomics and so on. And 77 is again a landmark. Gilbert and Sanger came together and proposed a method, or established a method to sequence the DNA. Until then we are talking about DNA as a nucleic acid without really having no tool to understand what is the sequence. Much of the protein sequence are known from protein sequence. But these are the people who came up with the method that we call as dideoxy sequencing, which is a very efficient tool for sequencing the DNA. They proposed and they established, they have shown that can be used, and that led to enormous data, because then every lab started sequencing DNA and RNA, because now you have a reverse transcriptase, so you can convert the RNA into DNA and sequence the DNA. So basically you can understand the sequence of RNA from DNA using the same method. That again is a seminal contribution. And last but not the least is the contribution from Carey Mullis. We used all this principle right from the 1956 Arthur Kornbank's principle that there are enzymes that copy the DNA, to the double helix concept and so on. What he came up with is a simple approach to make millions of copies of a small segment of the DNA using an enzyme, DNA polymerase, and the 4 bases of the DNA, and you can make in a tube quickly in 5-6 hours enormous copies. This method has really changed the way you look at the DNA, because earlier the people used to put the DNA inside a host, for most often E. coli, and allow the E. coli to grow and make copies on its own way. So it has removed all these bottlenecks, and you are able to make millions of copies of a small segment, so which is now being used every day in clinic, in labs, for diagnosis, for any other such kind of approach. So in that way, the contribution that I talked about from 1956 to early 1980s is something which you call as molecular biology era, because they were able to understand, analyze and manipulate the DNA, and that led to the field what is called as genomics, and that we will see in our next class.